So, hello students, I'm back and I'm in my new office. My house has 8 people in it and it's really busy, so I decided to come to a neighbor's house. They are on vacation, so we figured this would be a great place for a couple of weeks for me to come and make videos and whatnot. So this is my neighbor's house that we're taking in their mail and doing stuff like that for them. So they said, oh, sure, you can come on over. They're an older couple on vacation in Florida. So here we are in this lovely house by myself, quietly enjoying the peace and quiet, frankly. In any case, we're going to talk about Chapter 15 in 3 parts. The first part will be mostly introduction, and then the second part will be mostly problem solving, and then there'll be some more information in Chapter 15 that I want to talk about. And we are going to skip quite a bit of Chapter 15. I'm going to pull up the PowerPoint for Chapter 15, and you'll see right off the bat that we are going to skip quite a bit of it. So Chapter 15, the chromosomal basis of inheritance, Part 1. We're going to have 3 parts. And there are a couple of things that you notice right off the bat. Imprinting and extranuclear inheritance, I cut those out. I left the slides here because, God, this is such interesting material. And if you want to, you can take a look at these. Read it in Chapter 15. It's just really stuff that I think students are inherently interested in. So I left them on there just because, if you're interested, take a look. We are going to omit a huge concept in Chapter 15, though, and that is the linked genes concept. All right, so here we go. In the 1800s, remember, in the middle of the 1800s, Mendel kind of came up with several laws of genetics that still hold true today. And he used the term heritable factors to talk about what we know as genes. But no one really listened to his story. No one really grabbed onto his ideas about how traits are inherited until late 1800s and the early 1900s. At this time, people were starting to look at chromosomes and look at what was happening to them in mitosis and then in meiosis. And researchers figured out that these heritable factors that Mendel talked about are actually located on chromosomes. And we call these heritable factors genes today. And it turns out that Mendel's 2 laws, his big laws of inheritance, make sense in light of the way that chromosomes move around with their genes on them during meiosis. And there's a great figure 15.2. It's on the next slide. We'll take a look at it. Here's a fun, I call this a dad joke. And what did Mendel say when he founded genetics? Whoopee! Get it? Peas. All right. Anyway, this slide right here also has, if you are interested, the Mastering Biology animation might be useful here. And this slide shows us a cross that we saw in Chapter 14 with peas, the yellow round crossed with the green wrinkled seeds. And we followed this cross for 2 generations. This was our dihybrid cross that we looked at. And what it shows is if we think about these genes as actually being on chromosomes, then we can see how Mendel's laws work. So in this picture, we've got these 2 parents that are homozygote, right? A homozygote dominant parent, that's yellow and round, and one that's homozygote recessive, that's green and wrinkled. Our first generation flies, our first generation peas all have yellow round seeds, but they're dihybrid. They're heterozygote at both genes. Now, if we follow this first generation plant as it goes through meiosis, that's when we can see the laws of segregation and the law of independent assortment. Remember that the law of segregation says, hey, when you have 2 copies of a gene and they separate during the formation of a gamete. So in our drawing here, we have 2 copies of the gene, right? We have a big R and a little r. We have a big Y and a little y. And they're on homologous chromosomes that pair up in meiosis 1 and then separate in anaphase 1 of meiosis 1. So that you end up with 1 copy of each allele in each gamete by the end of this cross. In meiosis 1, there's also evidence for the law of independent assortment. Now, if we've got two genes that we're studying seed shape and seed color, and it turns out that these genes are on different chromosomes. We've got the yellow round, sorry, I forget what these are. These are the wrinkled or the round, the round or the wrinkled alleles on the large chromosome, and the yellow or the green alleles on the small chromosome. And the law of independent assortment says that alleles of genes on non-homologous chromosomes, that is, the R's and the Y's are not on homologous chromosomes, they're on different chromosomes. These alleles assort independently. In metaphase 1, the way that a pair of chromosomes lines up is independent of the way the next pair lines up. So in our drawing on the left, we see that the big R and the big Y ended up on the same side of the cell, and the little r and the little y ended up in the same side of the cell. Over here, the little r and the big Y ended up on the left side of the cell, and the big R and the little y ended up on the left side of the cell. So as this cell goes through meiosis 1 and meiosis 2 and becomes a gamete, we have gametes that can either be big Y, big R, little y, little r, big Y, little r, or little y, big R. And each of these are equally likely. Now, if you do a giant Punnett square, you'll end up with a 9:3 to 3:1 phenotypic ratio from this dihybrid cross. Okay, so what we're going to do in this chapter is think about genes being on chromosomes and the evidence for that and some different features that we understand about genetics when we think about genes as being on chromosomes. So let's move on. I am having trouble moving my slide. Page down. Something is odd. Let me work this out here. We are seeming to be locked in this. Okay, let's work it out. Let's move down. Who knows what happened there? Moving on. So the person that we want to talk about that helped us understand that genes are on chromosomes is Thomas Hunt Morgan. He worked with fruit flies. Early 1900s, fruit flies are a great study organism because they're small. They grow and reproduce really quickly. You can grow a lot of them in a tiny space, and they have really good definable characteristics that are easy to see and easy to study. And so fruit flies have been worked on for over 100 years with respect to genetics. Anyway, he did this early experiment with fruit flies, that's Drosophila melanogaster, that provided us with evidence that genes are on chromosomes. He decided to look at an eye color mutation in fruit flies. So most fruit flies have a reddish-brown eye. We call that the wild-type eye color because it's the color that's typically seen in the wild. Other flies have this white eye. They're a white eye mutant, if you will. And so Thomas Hunt Morgan decided to work with these and to try to sort out how this trait is inherited. So he did a number of crosses. This is just one of his crosses, but this is what he did. And we've got our parent generation, our first generation, and our second generation listed here. These are our cute little parents. Our parents are true breeders, okay? They come from a true breeding stock, if you will. He decided to cross a white-eyed male with a wild-type female. And he looked at the offspring. After the first generation, all of the offspring had wild-type eyes. Okay, that makes sense. Probably the mutation is recessive, and the wild-type allele is dominant. Then he decided to mate siblings, so he did an F1 by F1 cross, and he looked at the offspring. And we would expect from a cross like this, if we were thinking about a single gene trait that's dominant or recessive, we would expect a 3-to-1 ratio, a 3-to-1 phenotypic ratio in the second generation. He sort of got that, but let's look. He found that all of the females are wild type, half the males are wild type, and half the males are white-eyed. We were expecting to see that 3/4 of the offspring were wild type and 1/4 of the offspring were white-eyed, and that's kind of what he got. You know, we can highlight this a second. This represents 3/4 of the offspring, right? And this represents 1/4 of the offspring. But what's strange about it is we've got males, only males that are white-eyed. Females are never white-eyed. So what's going on? The sex ratios, the sexes are unusual. Morgan knew something about fruit flies. It turns out that a lot of people had been studying fruit flies already and knew a lot about their chromosomal makeup, and they were starting to understand meiosis a little bit too, right? So researchers knew, and so did Morgan, that male and female fruit flies have 4 pair of chromosomes. We have 46 chromosomes, or 23 pair. So fruit flies have 4 pairs of chromosomes. That is 8 total chromosomes, right? 3 pairs are autosomes. That's a new word for us. Those are chromosomes that are similar in both males and in females. So if we look at this picture down here, this is a picture of chromosomes in a female fruit fly on the left and a male fruit fly on the right. And the female and male fruit flies have 3 pair of chromosomes. So there's 1 pair, 2 pair. Those little guys are pair 3, 1, 2, 3, that are the same in males and in females. And then they found a pair of chromosomes that were different in males and in females. Females have 2 chromosomes that look alike. Those were called the X chromosomes. And males have 1 X chromosome and then this other chromosome that looks different, and they called that the Y chromosome. And so Morgan knew this, and Morgan said, I wonder if genes are located on the chromosomes, and this particular gene for white-eye, maybe it's located on the X chromosome. This is Morgan's explanation. So he said, perhaps the gene that results in the white-eye character is on the X chromosome. We know that males and females inherit different numbers of X chromosomes. Females inherit 2 X chromosomes, and males only inherit 1. So that could change the pattern of inheritance between males and females. Let's check it out. Let's see if Morgan's hypothesis makes sense. So we're going to draw out Morgan's cross between his white-eyed male and his wild-type female, and we're going to draw out the 2 generations of that cross. I'm turning my notes here. Okay, so what we're going to do is we're going to think about this gene for white-eye as being, or for eye color, as being on the X chromosome. And we're going to use the nomenclature that fruit fly geneticists use. So on the left here, I've already written it on my screen. I can't seem to get up a, oh, there we go, maybe. Oh, it's a tiny little red dot. You probably can't even see that. But up on the left corner, I've got little w is the allele for white-eye, and little w with a superscript plus sign is the allele for wild-type. This is how geneticists like to write it. The wild-type allele always has this superscript with a plus sign on it. So you take the name of the mutation and you put a superscript on here. That means it's the wild-type. And if the allele, the mutation is recessive, we use a small or a lowercase letter. Okay, so now let's think about this for a minute. Our white-eyed male has 1 X chromosome and a Y chromosome. And he's white-eyed, so he must inherit a white-eye allele. I'm going to put it on the X chromosome, like as a superscript, so that you can see it's on the X chromosome. And he has a Y chromosome. All right, that pairs up with the X chromosome. The female, who's wild-type, is a true breeding wild-type-eyed female. So she gets the 2 copies of the gene for wild-type color. Okay, does this make sense? Now, when we look at what the first generation inherits, we have to think about males and females. Okay, the first generation females, let's think about the females first. They are going to inherit an X chromosome from dad and an X chromosome from mom. So let's put these, actually I think I'll put mom's chromosome first. I like to put wild type and alleles first just because it makes it easier. This female should be a heterozygote, and she should be wild type in color. She is. The males will inherit, the males to be male have to get a Y. So they're going to inherit a Y from their dad. The Y doesn't have any genes on it. And they're going to inherit one of these X with a wild type allele from mom. I'm going to write anything that's a sex-linked trait, like this is, as being a superscript on the X chromosome. Notice that the Y chromosome doesn't have anything. So does this make sense so far with what Morgan got? Remember, Morgan said that all the first generation were wild type. Let's see. Is our first generation female wild type? Yes. And our first generation male wild type? Yes. The female here is heterozygote. The male only gets one copy of the gene. And so is it homozygote or is it heterozygote? It's neither of those. It's hemizygous. We would say that the male is hemizygous. You can add the zygous to it, even if you spell it wrong. It's okay. So now we're going to look at the second generation to see if we get the same result as what Morgan got, right? Let's take a look. Now, we might have to draw a Punnett square for the second generation, and I'm going to have trouble fitting it in, I think. You guys might have to do a better job than me. I'm going to draw in my Punnett square. Now, when this female, who's heterozygote, makes an egg, her egg is either going to have the X with the wild type allele, or it's going to have the X with the white eye allele. The male is going to inherit, or it's going to donate either the X with the wild type allele or the Y, right? So the two boxes on the right in my Punnett square are going to be males. The two boxes on the left are going to be females. And I just need to fill these in a second. Let's fill this in. Hopefully we can read it. You probably are doing a little better job. It's really hard to do this on my screen with my finger is what I'm using. Okay, let's see if this fits with what Morgan found. Remember, Morgan found that all the females were wild type. Let's check that out. Are all the females wild type? The top box is a wild type female. The bottom box below it is, oh, it's a wild type female who's heterozygote. What about the males? Morgan found that half the males were wild type and half the males were white-eyed. Well, the top box represent males that are wild type, and the bottom box represent males that are white-eyed. Hey, guess what? Morgan was right. Morgan identified that this mutation must be on the X chromosome because the pattern of inheritance fit with the idea that this gene is on the X chromosome. Yay, we've got a sex-linked gene. So what did we conclude? Now this is a slide that looks pretty similar to what I just drew. It's a slide representing the crosses of those three generations, the parent, first, and second generations. And the only thing that's weird about it, I find kind of oddballish, is these like creepy looking eyes or heads of fruit flies. Anyway, this is the same cross we just did. And here's what I want you to note. Okay. Remember, males are inheriting only one X chromosome and only get one copy of all sex-linked genes. And what's nice about Morgan's work is that this demonstrates two things. It tells us that genes are probably on chromosomes. It fits with that hypothesis at least. And secondly, it gives us an example of a sex-linked trait or a sex-linked pattern of inheritance. So it was a great study. Yeah. All right, so now we've got sex-linked sex linkage, we would call it. And we can talk about genes that are located on the X chromosome as being sex-linked genes. Okay, that's fruit flies. What about sex linkage in mammals? Let's check it out. So, do mammals have sex-linked traits? Absolutely. Let's look at humans. We're looking at a karyotype on the right of a human that has 22 pairs of chromosomes that are just called autosomes because they're the same in males and in females. And a pair of chromosomes we call the sex chromosomes that differ in biological females versus biological males. A biological female will inherit 2 long X chromosomes. A biological male will inherit 1 X and 1 Y chromosome. Now I'm going to show you a picture of the genes that tend to be on these chromosomes, but there's the X and the Y. This slide here shows us that the X chromosome has many more base pairs, that is more nucleotides, and many more genes, maybe around 2,000 genes on the X chromosome. The Y chromosome that males inherit has, very few genes on it, relatively speaking. One of the genes that's important, they're all important. One of the genes on the Y chromosome is called the sex-determining region Y. That's called the SRY gene. And this gene contributes to the development of the testes in males or in fetuses that have the Y chromosome. And as the testes develop, the testes produce testosterone that leads to the phenotypic development of a biological male. Okay, so we also have differences in our sex chromosomes. A couple of questions down here that we want to take a look at. Are the X and the Y chromosomes fully homologous? What that question is asking is, do the X and the Y chromosome pair up at meiosis? No, actually, that's the second question. The first question says, are they fully homologous? That means, do they have all the same genes? No, we just saw that. No, they're not. The Y chromosome is much smaller with many fewer genes. But do they pair up during meiosis? Yes. The X and the Y chromosome pair up in males during meiosis. They are partially homologous. So they pair up and then separate into gametes when the male is making, why do I keep saying the male? Well, the male makes sperm, and sometimes the sperm will get an X, and sometimes the sperm will get a Y. When the female makes eggs, the eggs all get an X. So the question I have for you is this third question. Does the male or the female gamete determine the biological sex of an embryo? This is like an exciting question. It's the male. The male's gamete determines the biological sex of an embryo, or the chromosomal sex of an embryo. That embryo inherits either an X from the gamete or a Y from the male gamete. So it's the male's sperm that determines the biological or chromosomal sex of the embryo. Okay, so a couple other things here. First of all, there's a comment that I'm making that is just relevant and really, it's relevant. We're learning today that the biological sex is not a binary condition, and I have a slide that you can go ahead and read on your own about what the term intersex means. Okay, it turns out that biological sex is pretty complex, actually. It's not as simple as, there's males and there's females, and we're really starting to understand and explore that a lot more in today's world than we were even 5-10 years ago. And I wanted to focus on the human disorders that are related to, these are disorders that would be called sex-linked disorders or sex-linked traits that are found on the X chromosome. Oh, I forgot one more slide in here. This slide shows us some different mechanisms of sex determination, because it turns out that sex determination in all different kinds of animals is variable. It's kind of fun and variable. We're here, that's why I circled it earlier. We are what we call the XY system. And we have, females have 2 X chromosomes, biological males have an X and a Y. They donate either an X or a Y in their sperm. Females donate an X, and together then we can get either males or we can get females, right? There are different systems for different animals. Some animals don't even have chromosomal-based sex determination. So if you look at reptiles like turtles, many of them have temperature-dependent sex determination. Temperature-dependent sex determination means when the female lays her eggs in a nest, the temperature at which the eggs incubate determine whether the turtles will become female or become male. And so it's not related to a chromosomal basis. It's the sex determination is related to temperature. So we don't have that. We have a chromosomal sex determination, okay? And here's that slide that you can take a look at if you want to learn a little bit more about the term intersex and what it means. Okay, here are a few X-linked conditions. By X-linked conditions, I mean these conditions are determined by genes that are on the X chromosome. There. So you maybe heard of many of these. I have, you've probably heard of Duchenne muscular dystrophy. Duchenne muscular dystrophy is a disease in which individuals lack the ability to make a protein called dystrophin, which is really important in maintaining muscles. And so people who are born with this, mostly boys, will slowly lose their muscles, and their muscles will deteriorate over time. And so a boy born with this will eventually end up in a wheelchair and will eventually die from this disease, probably in teenage or early 20s, something like that. Becker's muscular dystrophy is also determined by a gene on the X chromosome. It's similar to, but not as severe as Duchenne's. And here's one that you may have. You might possibly have red-green color blindness, and if you do, you have the inability to see certain colors. There are several different types of color blindness, and several of those are determined by genes on the X chromosome. Males are much more likely to be colorblind than females. You can check this out. If you can't read the number in the picture here, you might have some trouble. You might be colorblind. There are a couple of hemophilias. Hemophilias are diseases in which you are unable to make a clotting factor, a blood clotting factor, and if you can't make that, your blood doesn't clot effectively, and that can lead to internal injuries. It can lead to internal bleeding. It can lead to wounds that don't heal properly, things like that. And there are 2 different versions, both more common in males. And then there's another, maybe you've heard of SCIDS, the condition in which you essentially have a total lack of an immune system, and that is also linked to a gene on the X chromosome. So these are just a few of the X-linked traits linked to genes on the X chromosome. I think that's where I'm going to end, yes, and we will start part 2 by rolling into a number of problems about sex linkage, and we'll go from there. All right, let me close out of here. I'm really struggling to get this place rolling here. Okay, I think we're good, right? So we'll see you in a bit.